Okay, so today I'm going to talk about the biosynthesis of the GPI-anchors and human diseases. Therefore, my talk focuses on the human GPI-anchored proteins. In the first part, I will give you the simple overview of the GPI-anchor biosynthesis. And in the second part, I will talk about two human diseases caused by the GPI deficiency. At least 150 various human proteins are GPI-anchored proteins. And human GPI-anchored proteins, of course, have the conserved glycan lipid core structure. And there are two possible glycan side branches. One is the N-acetylgalactosamine linked to the first mannose. And this N-acetylgalactosamine can be further elongated by galactose and sialic acid. Another possible side chain is the addition of the fourth mannose to the third mannose. So these side branches introduce the structural heterogeneity into the human GPI-anchor proteins. However, the biological function, significance of those side branches are totally unknown at the moment. The function of the protein parts are various. There are many cell surface enzymes, such as alkaline phosphatase and other hydrolytic enzymes. There are many receptors, adhesion molecules, and complement regulators. And complement regulators is one of the topics of my talk. And the prion proteins and others are GPI-anchored. Because the GPI anchoring is critical for the surface expression of those 150 human proteins, GPI biosynthesis is essential for embryogenesis, neurogenesis, immune system, and fertilization. And this shows how GPI-anchor proteins are generated in the cell and how it is transported to the cell surface. The GPI biosynthesis occurs in the endoplasmic reticulum, in the precursor proteins, and the GPI glycolipid are separately synthesized on the ER membrane. And the precursor proteins are translated and transported into the ER lumen, and it has a GPI-anchor attachment signal sequence at the carboxy terminus. And the GPI glycolipid is synthesized from the phosphatidyl inositol by the stepwise addition of the monosaccharide and other components. And this pathway includes the 11 biochemical reaction steps. And pre-assembled GPI is attached to the carboxy terminus of the precursor protein. And these nascent GPI-anchor proteins further undergoes the maturation reactions on the way its transport from the ER through the Golgi to the cell surface. There are several maturation reactions. And finally, on the cell surface, GPI-anchor proteins clusters, as you heard from the G2. And some of the GPI-anchor proteins can be cleaved within the GPI-anchor by the GPI-aces and shed to the extracellular environments. So if we look at the biosynthesis pathway more closely, and this pathway requires more than 20 gene products for the biosynthesis of the GPI and its attachment to proteins. So these are the PIG genes involved in the synthesis and transfer to the protein. The attachment of the GPI-anchor to the protein is mediated by the GPI transamidase. And the human GPI transamidase is a complex of 5 proteins, PIGK, GPA1, PIGST, and U. And the GPI-anchor protein precursor has the GPI attachment signal sequence at the carboxy terminus. And this signal is recognized by the GPI transamidase. And one of the components, PIGK, is a system protease-like protein. It has the catalytic cysteine. With this cysteine, the peptide bond between the omega site amino acid and omega plus 1 amino acid, this peptide bond is attacked. And then the signal sequence is cleaved off. Then the PIGK makes the substrate enzyme intermediate, which is linked by the thioester bond. And this thioester bond is now attacked by the amino group of the terminal ethanolamine on the GPI. And by this, the nucleophilic attack, GPI anchor is finally attached to the omega site amino acid. So the GPA1 protein is a peptide synthetase-like protein. So it is thought that the GPA1 mediates this final reaction. So the GPI attachment to the protein is the transamidation reactions. The nascent GPI anchor proteins, still the GPI anchor is immature. And the first reaction is the inositol deacylation mediated by PGAP1, as we heard from the Mabry. And the action of the PGAP1 is coupled with the folding of the protein part. So after the protein folding and the inositol deacylation, it is further processed by the PGAP5 mediated removal of one of the phosphate anolamine linked to the second mannose. Now the GPI structure is compatible, has high affinity for the KAG receptor. And the KAG receptor is a complex of the 4 P24 proteins. By the action of the KAG receptor, GPI anchor proteins are incorporated into or recruited into the COP2-coated transport vesicles. Then GPI anchor proteins are transported to the Golgi. And still the phosphatidyl lipid portion is immature, as you heard from the G2. Then the fatty acid remodeling occurs. And the PGAP3 is a GPI-specific phospholipase A2 that removes the unsaturated fatty acid, such as the arachidonic acid. Then to the lysyl intermediate, the saturated chain stearic acid is added back. And for this deacylation, PGAP2 protein is involved. So now there's the lipid structure. Lipid has both two saturated chains, which is compatible with the Rafter association and clustering. And some GPI anchor proteins gets the NS2 galactosamine side branch after fatty acid remodeling. For this addition, this the PGAP4, which is a recently identified NS2 galactosamine, the transferase. And the enzymes for the galactose transfer and cyclic transfer are currently unknown. And then the GPI anchor proteins are expressed on the cell surface. So this is an overview of how the GPI anchor proteins are generated in the human cells. Now I move to the disease, abnormalities in GPI biosynthesis. The complete deficiency of the GPI anchor biosynthesis doesn't cause disease, it causes embryonic lethality. And this is shown by the PIGA gene knockout mice. It is not surprising because 150 human cell surface proteins require a GPI anchor. So it's the complete deficiency causes the early death in the embryogenesis. But the incomplete deficiency causes the diseases. There are two examples of the GPI deficiencies in human diseases. One is the paroxysmal nocturnal hemoglobinuria, or PNH. And this is an acquired disease of adults. And this is the deficiency is caused by the somatic mutation in the PIGA gene. And the second disease is inherited GPI deficiencies, or IGDs. This is the inherited diseases of the children, kids. And this is caused by the hypomorphic mutations in one of the PIG or PGAP genes. And those mutations are inherited from the parents. So I will first describe the PNH. So the PNH is an acquired disease. The number of patients is about 1 in 100,000 people. So I would expect in India at least 10,000 patients with PNH. And this is caused by the somatic mutation in the PIGA that occurs in the hematopoietic stem cell. And PIGA is an X-linked gene. So in the hematopoietic stem cell, in case of male, of course, there is only one PIGA gene, one X chromosome. So the one somatic mutation, one hit, immediately causes the GPI-anchor deficiency. So the GPI-anchor pathway is quite sensitive to the somatic mutation. This one hit causes the deficiency. In case of the female cell, female has the 2X chromosome, but one of them is inactivated in early embryogenesis to have the X chromosome gene expression level similar to the male. So even in the female stem cell, there is only one PIGA allele. So if the somatic mutation hits this active allele, then again they immediately cause the GPI deficient phenotype. So because of the X chromosome location of the PIGA, one hit makes the mutant cell. So what is PIGA? PIGA is involved in the first initial step of the GPI biosynthesis. This reaction is a transfer of the N-acetylglucosamine to the PI. So the PIGA is a catalytic subunit of the complex, the enzyme protein complex, which is the N-acetylglucosamine transferase. So in the affected PNH cell, PIGA doesn't function because of the somatic mutation. So the pathway doesn't go. So then in such the condition, GPI transamidase doesn't remove the carboxy-terminus hydrophobic signal sequence. So the precursor, such precursor is eventually degraded intracellularly, and most likely by the ER-associated degradation system. So there's the PNH cell, the cell surface expression of GPI-anchor proteins are gone. And for this pathway, there's more than 20 PIG genes are required. But as a PIGA mutation accounts for the most of the patient, the GPI deficiency. That's because all other PIG genes in this pathway are autosomal genes. For the autosomal genes, such as PIGB, PIGC, two alleles are active, the maternal allele and the paternal alleles. So to have the GPI deficiency, two somatic mutations in single cells are required, but such frequency is extremely low. So practically, the only PIGA mutation can be responsible for this acquired disease. So because of the somatic mutation in PIGA, the mutant cells have the GPI-anchor protein deficiency. In the blood cell gets the abnormality. So the GPI protein in the blood cell contains two important complement inhibitors, CD59 and DAF, decay accelerating factor. So the mutant stem cell generates large number of clonal abnormal blood cells, including red blood cell, deficient in the complement regulatory proteins. Due to the deficiency, the affected red cells are very sensitive to their own complement. So the patients get a complement-mediated hemolysis and hemolytic anemia. They also get venous thrombosis and bone marrow failure. So this shows the surface expression of DAF and CD59 to GPI-anchored complement regulator on red cells. So all the normal red cells express both inhibitors, but in the PNH patient have two populations of red cells. The normal population which express DAF and CD59, the other abnormal clonal cell population completely deficient in CD59 and DAF. So those abnormal red cell population are not protected from the complement. So they are sensitive to complement. And this illustrates how the intravascular hemolysis occurs. See, the patient blood is a mosaic. They have the normal cell population and the GPI deficient cell population. The PNH cell don't express DAF CD59. And when the patients get infection, for example, complement is activated in the body, then the sum of the activated complement fragments become bound to the red cell surface. So this is an inherent risk of the complement system. The some activated complement attaches to your own cells. But in the normal red cell, they have the complement inhibitors. So that they immediately inhibit the active complement fragments. But on the PNH cell, there is no regulation due to the deficiency. So this reaction goes on. And finally, membrane attack complex of the complement is generated. This is a channel. Then water comes into the red cell. Red cell become the balloon and the burst, and the hemoglobin is released. So it's called the hemolysis, complement-mediated hemolysis. And the hemolysis of the abnormal red cell population occurs at once during the infection. So the patients get a hemolytic attack and a severe anemia. So this is one type of the GPI-anchor deficiency. This is acquired disease caused by the somatic mutation. Now I go on to the second disease, which is inherited disease of the children. And this is a list of the disease-causing mutation in the cases of the inherited GPI deficiency as of the November last year. And already the disease-causing mutations are described, reported in the 17 PIG or PGAP genes. In total, the 170 patients with the IGDs are reported. And the number of patients is growing. So the discovery of the mutation in the GPI pathway this is the thanks to the whole exome sequencing that is used for the rare genetic diseases. That started about 2010. And so I will introduce the first report that studied the mutation in the GPI pathway. The Peter Kravitz in Germany used the whole exome sequencing and to the children with a hyperphosphatasia mental retardation syndrome. It's a rare genetic disease. And they found mutations in the PIGV genes. And they found basically the base substitutions that cause the amino acid substitutions in the conserved amino acid residues in the PIGV gene. PIGV is the second mannose transferase that are the second mannose to the GPI. So the Peter Kravitz asked me to test whether the mutation that found in the patient actually causes the reduction in the PIGV function. So we collaborated, and we used the PIGV defective Chinese hamster ovary cells as the test cells. And this is the result with one of the mutations, it's alanine 341 to the glutamic acid. And the PIGV defective CHO cells doesn't express CD59 or DAF GPI-anchor proteins. But if you transfect wild-type PIGV cDNA, the surface expression of GPI-anchor proteins are fully restored. But if we transfected the mutant cDNA, the surface expression recovery is very weak. So apparently the mutant has some activity, but is severely reduced activity. So if we use the stronger promoter, the recovery by the mutant cDNA is much better, but still this is subnormal level. And Western blotting detection of the PIGV protein is the mutant protein level is very low compared to the wild-type cell, wild-type PIGV. So we concluded that this mutation caused the instability of the PIGV proteins and reduction of the total activity in the cell. So what is the hyperphosphatasia mental retardation syndrome is also called the Mabry syndrome, which is reported by the Dr. Mabry in 1970. And 2010, the two groups revisited the Mabry syndrome. And biochemically, Mabry syndrome is characterized by the hyperphosphatasia, high alkaline phosphatase in serum. The alkaline phosphatase is normally the GPI-anchor proteins. And the primary defect of those affected kids are the retardation or slowed the development, the mental development, and they have the seizures. So that's the primary defect of those patients due to the partial deficiency is neuronal deficits. And some have the brachytelephalangy. It means that the defective, the formation of the last digit of the fingers. So in this case, is the last digit of the fifth finger is missing. So this also this trained pediatrician can pick up the characteristic facial the properties of the kids, and they say this the distance, wider distance between eyes, and the unique the form of the nose, and the unique the mouth. And they say the tented mouth, and this is the hyperphosphatasia mental retardation syndrome. So if we study the cell surface expression level in the patient blood granulocyte, the reduction is very mild. So there's compared to the normal level of the GPI, and the patient granulocyte has the partial deficiencies, the reduction. And reduction is very mild. And the CD16 is one of the GPI proteins is the most sensitive. They decrease. But the CD59 is not decreased. So it is the partial reduction of the GPI formation. In some GPI proteins are affected stronger. Some is the more resistant. So because CD59 level is normal, those kids don't get the hemolysis. Mainly is a neurological problem. Since the biochemical feature of the Mabry syndrome is hyperphosphatasia, so we studied the mechanism of why does the alkaline phosphatase is secreted into the blood. So we did use the model system, again the PIGV defective CHO cells, and we used the placental alkaline phosphatase. Alkaline phosphatase is all GPI-anchor proteins. And if you transfect the placental alkaline phosphatase cDNA into the PIGV deficient CHO cell or the PIGV rescued wild type cell, so the alkaline phosphatase is expressed on the cell surface on the wild type cell very well. But in the PIGV knockout defective cells, this surface level is very, very low. But if we measure the phosphatase activity in the normal cells, most of the alkaline phosphatase activity is in the cell lysate associated with the cell. Only small activity was found in the culture supernatant. But in case of the PIGV mutant, most of the activity was found in the culture supernatant. The cell-associated enzyme activity is very low. So there's a lot of GPI, the alkaline phosphatase is secreted into the medium. So we purify the secreted GPI alkaline phosphatase from the culture supernatant and determine the structure by the mass analysis. And we found that the alkaline phosphatase is lost the GPI attachment signal. So the attachment signal is removed. And the C-terminal was the omega site serine. So this is our view, how does the alkaline phosphatase is secreted. In the wild type cell, the GPI-anchor attaches the intermediate and the proteins anchored and stays on the cell surface. But in the PIGV deficient conditions, the secreted GPI-anchor, secreted alkaline phosphatase has the omega site at the C-terminus. So we think the transamidase undergoes the first reaction, remove the signal sequence. And this intermediate cannot be attacked by the GPI, because the GPI is truncated, there is no amino the group to attack the intermediate. But this is a thioester-linked intermediate, which is eventually can be hydrolyzed by water. And now this soluble alkaline phosphatase generated. This can be secreted into the supernet. So we think that this is the mechanism of the hyperphosphatasia in the patients. So I will stop there. I would like to thank my colleagues in Osaka, and I would also like to thank the group in Germany for the collaboration in the IGDs. So thank you very much for your time.